Assalamualaikum everyone, welcome back to this channel. So this is me Ali Wahid and you are on the channel of Medicare. So in this video I am going to start the chapter number 2 of pharmacology. So in the previous lecture we have discussed all the things about the chapter number 1. We have covered the whole chapter of pharmacokinetics. We have studied all the types of the actions of your body onto your drugs, that your drug is how the drug is absorbed, how it is distributed, how it is metabolized, and how at the last it is eliminated out. Now, in this lecture, I am going to start another branch of pharmacology, and this branch is called as pharmacodynamics. So, always, first of all, we are going to discover what is the definition of it. So, always, I am going to define, understand you the definition of it in the form of various parts. So, divide the word pharmacodynamics into the two parts. So, it consists of two parts. First part is pharmaco and second part is dynamics. So the word pharmaco means the drug which you are giving to someone, and the dynamics mean the movement and the actions. So in this type or in this branch you are going to discover that what kind of actions of your drug onto your body. So आपका आपकी drug, what the drug is doing inside your body. So now let's discuss that. First of all, after entering into your body, it will goes to the specific type of receptors on which it attacks and produce a particular kind of response. So just take an example that this is a membrane and on which a receptor is present. Now what will happen, you take the drug and your drug will enter into your body, will attach to that receptor, this receptor become activated, and then it will produce particular type of response which is required for it. So now let's see that what is a receptor, okay, onto which your drug is going to bind. So one thing you have to remember that whenever you eat a drug or take it, like for the hypertension, you eat a drug which decrease your blood pressure. So never it happens that your drug is going and produce a magical effect and you will treat it out. Never it happens. Always a significant type of mechanism will happen. The drug is bind to a specific receptor and after that it will produce a particular response which is required. So now let's see what is the receptor. So receptor is any drug or the substance which is present in the body bind to a specialized target macro molecule. Macro mean big molecule, which is called as a receptor. So what are the examples of receptors? So your receptors can be intra available inside your body or can be from the outside, like the drug which you eat is the exogenous ligand, and some substances are also present inside your body like enzymes, like protein, like nucleic acid. They can act as a receptors in your body. So like enzymes, you know very well that they have specific kind of binding site on them, and this which is called as the active site on which specific kind of substance is going to bind and then it will perform its action. So they are the types of receptor actually. Now what happens after the binding or between the drug and the receptor? So a complex will form which is drug receptor complex and which could be ligand receptor complex. So your drug will combine to your receptor and form dr is the drug receptor complex, and your ligand react with receptor and form lr which is ligand receptor complex, and this change is reversible. Okay, it can goes from here to here, from here to here. So therefore there is establishment of equilibrium between them inside our body, and whenever this equilibrium disturbs, then some types of diseases can happen to us. So now let's discuss what are the states of receptors. So your receptor is always in two states, whether it is in the inactivated form and whether it is in the activated form. So how it is activated? When a drug or a ligand bind to it, then then it will change from its inactivated form into the activated form. And whenever your drug or ligand removes or move outside or get detached, then it will again convert it itself into the inactivated form. So these cycles changes in your body, and various kind of functions or metabolism is happening inside your body. So let's discuss what are the types of receptors first, then we also discuss what are the types of ligands which are present in your body. So actually there are two main types of receptor which are present. First is the intracellular receptor. The word intra mean inside, cellular means cell. So these are those receptors which are present inside a cell. So there are so many receptors which are present inside, like suppose this is a cell. So there are some receptors which are freely floating in the cytoplasm, and like a ribosome is a receptor and it's freely floating in your cytoplasm. And some receptors are present on the membrane of your nucleus also. So the drug has to cross the lipid bilayer and has to attach with that receptor and then to produce the particular response. Now, the other type of receptor is the extracellular receptor. Extra word mean which are the outside of your body. Cellular means cells. So they are these receptors are present on the membrane. The drug has to bind with them and produce the particular response. So they are the two main types. Now, the extracellular receptors are further subdivided into the three types. Here are the three types. The first type is the ligand gated ion channels. These are the types of a channel. And other are the G protein coupled receptors, and the last is the enzyme link receptors. Now we are going to discuss the detail of all of these receptors, and we discuss that what are the mechanism of their actions. So here I have explained all of these receptors here. So first of all, see on this diagram. So the first receptor which I have drawn here, this is the ligand gated receptors. So this is the receptor activated site on which your ligand is going to be bind. So this is the activated site. Now I am drawing the ligand here, which is of its own shape. Now they will bind with it and perform the specific reaction. On the binding of it, what will happen, this ion channel opens. Now it is closed. Now after the binding of ligand, it will opens itself like this. Now this channel is opened, as you can see here. Now the ions like suppose it is wants the diffusion of sodium inside, so now it will allow the sodium to move inside. So this is the ligand gated channels, which is the gated mean closed ion channels which opens itself on the attachment with the ligand. Okay, and how much their duration is, so they perform their actions within the few milliseconds. What are the examples of them? So the nicotinic receptors which are present in the somatic nervous system. You know very well about the somatic nervous system, that it is a nervous system which is not automatic. It is attached on the skeletal muscle. So let's suppose this is the nicotinic receptor which is attached on your skeletal muscle. So this is your muscle. Okay, so now what is going to be happen, this is a post synaptic neuron. So this neuron is releasing the nicotine, you can say nicotine, you can say acetylcholine. Now what will be happen, this acetylcholine bind with this nicotinic receptor and causing the sodium deflux. So actually this receptor is just like this will cause the diffusion of sodium inside. Now what will happen, the sodium will move inside and the potassium will move outside. Now due to this movement, your muscle is going to be contracted. So these are the ligand gated channels which are present in your skeletal muscle and causing, and they are responsible for the contraction of your muscle. Now now there is the other type of receptors. This is the G protein coupled receptor. Coupled means they are binded, they are coupled, they are present with what, a G protein. So G protein is a type of protein which is coupled with this receptor. Okay, now Here you can see in the diagram, this is the G protein coupled receptor. This is the 7 cross membrane receptors. The binding site, which has to be bind with the ligand or drug, is present outside the membrane. Now the drug is going, or the ligand is going to be bind with it, and after the binding, this receptor is attached with the protein. This is a G protein. This is a G protein. Okay. Now, this G protein has three subunits attached with it. This G protein is consist of three subunits. Like suppose this is the alpha subunit, this is the beta, and this is the gamma subunit. This is beta and this is gamma. Now, after attachment of the ligand, this beta and gamma detach, and the alpha subunits detaches with itself. Now, this alpha subunits is attached with the GDP. Now, after the attachment of the ligand, this GDP phosphorylase, and it will convert itself into GTP. And GTP will helps to convert your adenosyl monophosphate into the cyclic AMP. And after the formation of cyclic AMP, there is activation of protein kinase, and your particular kind of cellular response will happen. So actually this is the secondary messenger system, which is happens inside your cell. Now there are three types of G protein, which is GS, GI, and GQ. GS means G stimulatory. That this kind of protein, when attached, it will stimulate particular action. Let's suppose this cell is responsible for the increase in the heart rate. So whenever the ligand is attached with the GS, then it will activate the heart rate and increase the heart rate. Second type of G protein is GI. So GI, whenever it attach, it will decrease the response. That it will decrease your heart rate. Now last is the GQ. GQ has some a different mechanism. So it is activating other types of your secondary messengers. Like it will activating, after the attachment, your GTP will activate phospho phospholipase C, and your phospholipase C will going to be produce two things, which is inositol phosphorylase 3, and a second thing is diacylglycerol. These two things will produce. Now the glycerol will produce the cellular response inside your cells. And your IP3, inositol phosphorylase 3, this is responsible for increasing the calcium intake, and whenever the calcium is intaken, the calcium calmodin and in secondary messenger system will activate it and perform particular kind of reaction. So this was the whole mechanism of G protein coupled receptor. And what is their duration? They act within seconds to minute. So the ligand gating channels are fastest, because they produce its effect in milliseconds. But the G protein takes sometimes, it can take time from some seconds to some minutes. Now let's discuss the examples of them. So various kind of muscarinic receptor. Muscarinic is a type of cholinergic receptor. So all the muscarinic receptors which are present, there are many types of muscarinic receptor, like M1, M2, M3. They are present in your glands, they are present on your muscles, they are present on your heart. So they are a G protein coupled receptor. And the next type of receptor is the adrenergic receptors. There are two types of adrenergic receptors, alpha and beta. So they are also G protein coupled receptors, and the alpha has further more types: alpha 1, alpha 2. Beta has further three types: beta 1, beta 2, beta 3. So you will discuss whole of this discussion in the next chapters, which we discuss in this session. So please, if you don't subscribe to this channel, then subscribe it, so that you can take all of these videos and can take benefits from them free of cost. Now let's discuss the third type of receptor which is present in your body, and this type of receptor is the enzyme linked receptor. As its name indicates, it is going to be linked, it is going to be attached, it is going to be coupled with the enzymes. and its duration of action is within the minutes to hours. Mean it is the most late type of receptor which produce very which is very lazy to produce its action. It can takes many hours to take the intracellular responses. Now let's discuss what is the examples. Various kind of insulin receptors are the examples of them. Now as you can see here in the diagram, so I am removing all of them, so in order to clarify the concept of this in your minds. Now what is going to be happen here? So this is an enzyme linked receptor. You can see here this receptor has two parts. Okay, this is composed of alpha subunits. So this unit which I am shading is the alpha subunits, and which is non-shaded part which become the beta subunit. So these two shaded parts are the alpha subunits, and they are joined with each other through a bond. So here I have found a bond. So this bond is the disulfide linkage. So disulfide linkage is this type of linkage is present between them, and the beta subunits and the alpha subunits are also attached with each other with the help of this disulfide linkages. Now what is happened, and this is the beta subunit. Okay, now these beta subunits are attached with the tyrosine. Tyrosine is the amino acid. Now what will happen whenever a ligand is came? Now let's suppose this is the ligand of this particular receptor. Now this ligand came, bind with it, and after the binding, what will happen? This tyrosine phosphorylase mean a phosphate group will attach with it. Both of these tyrosine phosphorylase. And the one thing which is explained in your book is that the both subunits attach themselves. Mean the first of all, the the left side is going to be a phosphorylase the right side. Now when the right side is phosphorylated, it will going to be again activating or phosphorylating the other side. So these both of them activated themselves, so this is called as auto phosphorylation. And why this happens and with the help of what will help to phosphorylase them? So there is an enzymes present which is called as tyrosine kinase. This enzyme will helpful in the phosphorylation. After this phosphorylations, the cellular response will happen and the particular kind of response will occur within a minute to hour. Now there is the last type of receptor which is the intracellular receptor which are present inside your cell. Now there is a serious type of confusion that how the receptor is present inside the cell and how the ligand is going to bind it. So that is clearly simple, that your ligand or drug which is present outside the cell, or it has to be diffused through the cell membrane, and after that it can go attach with some types of protein, and after that it will go into your nucleus, attach with the receptor which are present on the nucleus, and causing the transcription or translation. What is transcription? The conversion of your DNA into the RNA. What is translation? Conversion of your RNA into the protein. So it is causing the gene expression, changing the genes, and producing its effects. So the gene is controlling everything. If a gene changes, then you can control the cellular responses. Now the ligand which has to bind with these intracellular receptor could be intracellular or extracellular, mean there is no restriction on the ligand. Just your ligand which can be present inside a cell. For example, this is a cell. Now there are some types of receptors or ligand which are present inside. They don't have to move from outside of the cell into the inside. Okay, so as this ligand, it has to come from the outside of the cell, but there are some ligands which are present inside. They can also bind with these receptors. And what are the examples of these intracellular ligands? So here the examples of them is given, like steroidal hormones, protein, enzyme, RNA, ribosome. They are present inside, but there are some extracellular ligands like steroidal hormones. They have to cross with themselves and then has to be binded. Okay, so there are some hormones which are present inside, there are some hormones which are present outside, so there is difference. Now let's discuss. Now we have covered all the types of receptors which are given in our chapter or in our book. Now we are going to discuss another terminology which is used here, and this terminology is the spare receptors. So spare word mean to which has nothing to work, which is free. Okay, now examples is given that some types of insulin receptors. So what is the function of insulin receptor, or what is the function of insulin? So insulin is released whenever the concentration of glucose increases inside our body. So what happened, it is controlling the concentration of glucose, it is decreasing the concentration of glucose in our body. So the insulin we going and bind with the insulin receptors, and the insulin only bind with the 1% of the receptor, while the 99% of all the insulin receptors which are present in your body are spare. Only the 1% is performing or controlling the amount of glucose in your body. But in the case of beta 1 adrenergic receptors, so they are the receptors which are present on the heart. Now for their working, about 90% of all the receptors has to be on working attached with the receptors, and they have to work. But only 5 to 10% of the receptors which are spare, which doesn't have to work hard for it. So this was the terminology, and this was all the concept about the spare receptors. Now I am going to tell another concept about the receptor, which is the desensitization, down regulation, or up regulation. Then after that we will discuss what are the types of ligand, and then we will end our this lecture. So here the concept of desensitization or definition of desensitization is written that whenever the repeated administration of our drugs happen, that you will take the drug after repeated dose after every 1 hour, what will happens, it will lead to change, it will diminish the response. Now your receptor will desensitize, they will never sensitize with their cell due to the high concentration. So this phenomena is called as tachyphylaxis. So this is really really important to make a concept of you. Now let's discuss what is the down regulation and up regulation. Down regulation is the process in which no receptor is available for a particular kind of response. As a result, no response will happen. So this is called as down regulation. So what is upregulation? So in the upregulation, you are increasing the number of receptor, you increase the response. They are sensitive to the agonist. Whenever they find the agonist, they will bind with them and perform the stimulation or perform the activation of receptor. While they are resistant to antagonists, they will never perform the opposite effect, they will never block the receptors. So this process is called as upregulation. Now let's discuss what are the types of ligands. So there are two types of ligands, endogenous and exogenous. Endogenous ligands are present inside your body. They are in factly produced inside your body. Some hormones and acetylcholine, which is a neurotransmitters, are the examples of it. But the exogenous ligands, they are not produced inside the body, they are given from the outside, and like the drug of acetylcholine. So acetylcholine is a receptor which is already present inside your body, but there is a drug which is called as acetylcholine which work very similar to acetylcholine, and this drug is called as exogenous ligand, which because it is given from the outside of the body, not produced inside. Now let's discuss the types of ligands which are on the base of their action, that what kind of action they have. Like some drugs have stimulatory action, they are going to stimulate something. Let's give the examples of them. So their example is like epinephrine drug is given to patient, it will increase your heart rate. Similarly, when the norepinephrine is giving, it is also increasing the heart rate. So they are stimulate something, they are increase something. Some are inhibitory action, they are decreasing the action. Like the beta blockers are given in their examples. So they are responsible for the decrease in the heart rate, they are decreasing it. So they are blocking something, inhibiting something. So some drugs have just supplementary actions. Examples of them are like iron supplement. So iron supplement is useful when you have anemia. So in the anemia you have deficiency of iron, you have decrease in the red blood cell due to decreased iron. So when you give them iron supplement, as a result their hemoglobin increase. Due to increase in hemoglobin, your rbc increase, your rbc, sorry for that, and as a result your blood is increased. Now there are some other types of ligands like cytotoxic effects ligands. They are decreasing the effects of cancerous cells. You can say they are the anti-cancerous drug, anti-cancerous drugs. Okay, so they are placed in that group. Last is the irritant effects. So irritant effects are the drugs which are decreasing the irritating effect in your body, like nasal nasal sprays. So they are given when you have some kind of irritation in your nose due to mucus and due to some kind of bacteria and viruses. So therefore these drugs are given to in order to decrease that irritated effect. So this was all about the today's lecture. I hope that you will understand this lecture, and I hope that I will try my best to make all of these concepts in your minds. And if you don't subscribe to this channel, then go press the subscribe button and also press the like button in order to get the notification of all the video which I uploaded. So thank you so much for liking this channel, for subscribing to me. Now meet you soon in the next lecture. Till up, Allah Hafiz.